put your slides up. So it is with great pleasure for me to introduce to you Dr. Kevin Nash. Dr. Kevin Nash is a member of our FIRE consortium, and Dr. Kevin Nash is the assistant professor in the Department of Molecular Pharmacology and Physiology in the College of the College of Medicine at the University of South Florida. Dr. Nash, his interest and his passion is in gene therapy. He is really integral in the development of viral vectors and bringing gene therapy to Angelman syndrome, and has a really, really strong knowledge base and interest and desire to see how we can get best distribution into our children's brains. So he is going to talk to you a little about what he's doing, how he's doing it, and where this is going in the future.
Thank you. Can everyone hear me okay? What's my pointer here? Now listen to it. Don't use this one?
Use this one.
Forward, back. Okay.
And then use the mouse instead, because on the live stream you can't see the laser pointer.
Okay. Okay, so today I was asked to give you a lecture, essentially. So I'm going to teach all of you and make you experts in gene therapy. Just so you know, I made the pop quiz at the end pretty simple, so you should be pretty good at it. So what is gene therapy? In simple terms, it's an experimental technique where we can deliver genes into an organism, okay? And specifically deliver those genes into a cell. And it may be that we're targeting certain cells within the body. And that can be critical because you don't necessarily want to do a treatment for the brain and have your gene go to the liver, because that's not going to be very useful. So we want to make sure that we deliver the genes, and we want to make sure we deliver them correctly, and we want to make sure that they work. So there's a lot of things that we have to think about when we're going forward with this. And essentially what you're doing is you're introducing genetic material that corrects a problem that's already there. And in case of Angelman syndrome, we have a lack of the UB3A gene, so we're going to try to put the UB3A gene back in there. I just wanted to go over this again to make it clear in your mind. So, what is DNA? Of course, we know that DNA is essentially the stored information that encodes for everything in your body. So it's the way of the cell keeping a record of how to make things, okay? So how to make your proteins. It's an instruction manual. And unfortunately, not everyone's manual has all the correct information, and not everyone's manual is identical, otherwise we'd all look the same. So the information you get is critical to the function of your cells. And so let me try the mouse here. No, I'll just do the laser pointer. So you have your DNA. This DNA encodes, and it translates into RNA. That's not what's really important here. What's important is that it makes a particular protein. So you have your genes in your DNA, and essentially each gene you have encodes for a particular protein. Okay? So if you have a problem with that gene, you have a problem with that protein. But the good news is we can isolate these genes. So we can take a good gene and hopefully put it into a cell. So this is kind of a simplistic overview. So gene augmentation is essentially what we're trying to do. We're trying to put a gene back in. So we have a cell that's non-functional because it's missing a particular gene. We can introduce that functioning gene. So we isolate the gene, we introduce it to the cell. Now we have a cell that's functioning normally. It can produce the protein that it couldn't produce before. So there's a number of different ways we can get that DNA into the cell. The first one is essentially we just put the DNA on the cell and hope that it gets in there. You can imagine that doesn't work very well. Other methods there's like physical methods we can use, and one of those is called electroporation, and essentially that's a small electrical charge that opens up the cell, allowing the DNA to get in. We have chemical methods as well, so we can take the DNA, we can cover it with different molecules, and these molecules have the ability to move into the cell. So these molecules essentially transported the DNA into the cell. Some of these can be very toxic, so it's not a very useful system for people. However, the most promising is for humans at least, is probably viral vectors. So to understand this, we really need to understand how does the virus work, okay? So if you look at the life cycle of a virus, it infects a cell. It then moves to the nucleus where the DNA is present. It unloads its DNA. And then it takes advantage of the cell so that it can produce its own proteins, make extra copies of its own DNA. Now it can make new versions of itself, and then it's released and passed on to the next person, and that's how we get the flu traveling around the world. So in gene therapy, we don't want to keep passing on these viruses to other people because that's not very safe. But we do want to take advantage of this part of the infection cycle. So essentially, if you look at a virus, it's a very unique way to inject DNA right into the nucleus of a cell, right where we need it to go. So we're going to hijack this system and take advantage of what nature's already done quite effectively. So just terminology in case I spit out some words. So gene therapy uses a modified virus. Okay, so it's not the virus that's going to cause infection in people. And it's often referred to as a recombinant virus. And essentially that just means man-made. And it's often referred to as a vector. Okay, so a vector for us means something that can transport something from one place to another. Okay? And of course, it's critical that we modify the virus so it doesn't cause disease, but it still delivers the gene. So we're hijacking that system in the most advantageous way. Sorry, is there a problem?
Your mic is picking up some interference.
Oh, okay. Sure.
Thanks.
So some of the things that we have to consider when we're making a viral vector or making this gene therapy. The first is that we want to remove all of the viral genes that we can, okay? We don't want to introduce anything that's going to cause a disease. We don't want to introduce anything that may cause an inflammatory response. So your body recognizes things as foreign. So if we can remove all of the viral genes that are there, we're not going to make something foreign in your body that your body's going to be, oh, I don't like this, and then attack it. We also have to make sure that, and by doing this, we make sure that it also doesn't replicate, so it doesn't spread. Okay? But we still have to make sure that it targets. So the biggest thing for us in the gene therapy field is safety. And we have to prove that everything is safe before we can continue. So we're removing all of the viral DNA. We have to replace it, in this case, with the UB3A gene. So if you imagine a virus as a sphere, a balloon, a ball. So it's a sphere, it's hollow on the inside where it contains the DNA. So if we take that virus DNA and we remove it, okay, now what we can do is place in the UB3A. So now we have a virus that is identical on the outside. It's the same sphere, but we have the UB3A gene on the inside. So now we have a virus that can infect the cell and deliver the gene. So we remove the viral genome, we put in our UB3A gene, we infect cells, that virus does what it does, delivers the gene to the nucleus, and now it's going to produce our UB3A protein. So now our cell should be functioning normally because it has the UB3A protein there that we wanted it all along. If we look at the different types of viruses, there's a number of different families that we use in research and also in gene therapy. There's what's called the Retroviridae family, and you may have heard of lentivirus. These viruses are actually very useful in some instances. They can actually integrate their DNA into the host DNA. Adenovirus is not used as much anymore because it does produce an immune response. So we don't really like this one. It is, however, very, very useful if you're trying to do gene therapy to cancer cells, which we're not doing here. Herpes, not used very much. The one that is emerging as the most promising is adeno-associated virus. And it's called adeno-associated virus because it was originally found as a contamination of an adenovirus preparation. So what's so great about AAV? So one of the things is that it is very safe. It doesn't cause disease in humans. So the normal wild-type virus, the AAV that's present, does not cause any disease in people. And in fact, most of you probably have it in your system and you don't even know it. It doesn't cause an immune response. Unlike some of the viruses that we do use, it doesn't cause a big immune response. So your body doesn't react to it. You don't have problems accepting it. We do remove all of the viral genes. So with AAV, we can remove all of the viral genes, so we're not delivering anything that may cause a problem. And of course, the biggest thing is that safety has been shown in a number of clinical trials. So there have been a lot of clinical trials using AAV, and it has shown to be very, very safe. And of course, for us, we can deliver genes to the neurons in the brain. So we want to make sure that we're using a virus that will deliver to where we need to target. And we get long-term protein production. So once we get the gene in there, it's going to be active for a long, long time. And they've shown that it can be active for at least 6 to 10 years. And this is in the study that Allison was talking about with the retinal degeneration. So some of the technical challenges that we're trying to figure out is what is the best way to deliver the genes to the neurons? And how do we target as many neurons as possible? Okay, so we have to globally get this virus to go throughout the entire brain. And also we want to make sure that we control the gene expression. So we want to make sure that we're not producing too much, we're not producing it in the wrong place. We want to make sure that we're controlling how much we're making so that we make functional protein and make cells happy again. So one of the ways that we can do this is gene replacement therapy. So what that essentially involves is targeting the brain, having the virus delivered to the entire brain, and then produce protein in every neuron. This one can be a little bit tricky because, as you can imagine, we have very large brains, and it's difficult to get the virus to spread through the entire brain. So we're trying to figure out how to effectively do that in the animal models so that we can scale up into humans. So this would be targeting the neurons directly. The other option is a protein replacement therapy. So here what you can imagine is we're delivering a protein to the body so that the cells will take up the protein, and then rather than taking up the gene, they take up the protein, and then they fix themselves that way. So what is one way we can do that? We can also do this with gene therapy. So what we'll do is we'll target a small number of neurons, and then those small number of neurons will start producing the protein. So we essentially have a factory of cells now that are producing the protein. And they're going to release that protein from their cells, and that protein will then diffuse, if my thing will play, oh well, it'll diffuse out to the rest of the brain. Okay? And that protein will then be delivered to all of the neurons within the brain. So we don't have to target all of the neurons with the virus, just a certain subset of them. Those ones become a factory. That factory then delivers the protein to everyone else. So the other thing we're trying to figure out is there an alternative to just directly injecting into the brain? And perhaps there is. There's a number of researchers that are working on AAV biology. And you can see here's just a list of some of the publications just from this year that are looking at targeting AAV to the brain. So trying to specifically put the virus spread throughout the entire brain. And this group here did a very interesting technique where they generated a new AAV virus that we can actually inject in the tail vein, and then that virus actually moves to the brain. So now we can inject intravenously, and that virus moves to the brain. You can imagine this is going to be much, much less invasive on patients. So it's going to be much easier to do an injection into the bloodstream than it is going to be injecting into the brain. So what does that look like? If we take a look here, here's some mice that I did injections with. And here's a control. You can see there's no staining for the protein that we delivered with the virus. Here you can see how it's very dark compared to this one. So we have a lot of expression just from delivering the virus into the tail vein. So this one here actually uses a promoter that will express in any of the cells. So it can express in different cells within the body. But what we can do is take advantage of what's called promoters. They're DNA segments that regulate the expression of genes. And each gene has one of these promoters at the start. So it tells the cell, oh, you're meant to be produced in the liver. You're meant to be produced in the heart. We can use one promoter that says, you're meant to be produced in neurons. So with this neuron-specific promoter, we can also see expression throughout the brain. So now we can deliver a virus into the bloodstream. That virus moves to the brain and hopefully only expresses in the neurons in the brain. And I blew this up a little bit here so you can see some of the different brain regions, the hippocampus and the cerebellum. And you can see all the dark staining here, all of these neurons that are expressing the gene that we delivered. So this is some of the things that we're working towards, trying to get the expression. We'll have to test a lot of this in the animal models, where we'll also be looking at the new animal models that we have and see how well we can correct the phenotype or the symptoms that those animals display. So hopefully I explained gene therapy well enough that you all understood. And if you have any questions, I'll take questions. Either I explained it really well or I scared you. Yes.
You said that once you injected, the gene carried once you injected, it lasts 6 to 10 years. Is that right?
Yeah. So let me repeat the question. He was just saying that when we inject the virus, it'll last 6 to 10 years. So it's difficult to know how long it will last, simply because you have to have an endpoint to a study. So in that particular case, it was done in dogs, and they were trying to fix the retinal degeneration. And they allowed the dogs to live. The study went for 6 years, and the dogs could still see significantly well. So it's not that it stopped after 6 to 10 years. It's just that the study had to stop at some point. So it's conceivable that it will be for the lifetime of the patient. No? Okay. Well, thank you.